Very good day to all. I'm Bin Chui here, and today I'm going to give a presentation on the effect of preservative loading on the antimicrobial activity of titanium dioxide photocatalysts. So this project was supervised by Dr. Pei Wen from CSC Screen Process Supply Primary Limited and also Dr. Siew Ling from Universiti Teknologi Malaysia. So let's just start with titanium dioxide. Titanium dioxide was studied and found that it can produce reactive oxygen species, which is superoxide radical and also hydroxy radical, upon UV light irradiation. So these are always generate will photodegrade harmful pollutants such as VOC, bacteria, and also viruses. However, there is cons of titanium dioxide, which is it can only be activated under UV light. So a solution for it will be metal doping of titanium dioxide, where it can activate the titanium dioxide under not just UV visible region and also visible light region. And furthermore, it can improve the inactivation performance against viruses and also bacteria. Amidst COVID-19 pandemic, CSC have found and produced a metal doped titanium dioxide which called FOCAP. This FOCAP was used as a biocide additive that added into textile screen printing paste and coated on fabric mask. Studies had found that with this FOCAT, the bacterial killing efficiency can go up to 99.9% against Staphylococcus aureus in a 24-hour light radiation. However, there is still a problem with this FOCAT, where the initial killing bacterial efficiency of it was low and it is low in the effectiveness. Before going into today's topic, let's just start with the common biocide in the industry. So this too is the common biocide that used in the industry, where on the left is the methyl isothiazolinone, which is called in short of MIT, and on the right is chloromethyl isothiazolinone, where in short is called as CIT. The mechanism of how this MIT and CIT biocide works is where first, this MIT and CIT biocide will diffuse into the bacteria through cell membrane with the simple diffusion process and also energy required process. After the diffusion, it will react with the nucleophile group of the cell component through the electron deficient sulfur from the sulfonamide group. And this will result in the inhibition of bacterial cellular activities in minutes times and so leading to the cell death. However, this MIT and CIT biocide have a drawback where if the MIT and CIT was too high in concentration, it will be harmful to human and environment. Furthermore, the possible emergence of bacterial resistance toward MIT biocide will also occur. Lastly, the MIT and CIT biocide is non-reusable. So, for this research, we will try to design the limit of biocide use, which is lower than 0.68%, so that it will not be harmful to human environment. Synthesis a new titanium dioxide metal biocide composite and design a novel photocatalyst for antibacterial. Let's just start with why MIT and CIT biocide. MIT and CIT biocide was studied and has shown that it had high antimicrobial activity. Besides that, this MIT and CIT biocide was also a common isothiazolinone biocide derivative. Also, the biocide was used usually in cosmetic, paint, ink, and other industries. Hence, in this study, we will try to introduce this MIT and CIT biocide into the FOCAT, since this MIT and CIT biocide was reported that gave effective against a broad spectrum of bacteria and also fungi. The three main objectives of this research study will be to synthesize titanium dioxide metal CIT-MIT composite, to evaluate the opacity of the prepared composite, and to evaluate the antibacterial performance of the prepared composite. Let's just move on with the experimental of this research. A textile screen print paste modified with metal doped photocatalyst FOCAT was received from CSC Screen Process Supplies Private Limited. This FOCAT is a water-based emulsion which consists of metal doped titanium dioxide for antimicrobial properties and also acrylate binder to enhance adhesion on coated fabric. Three different loading concentrations, which is 0.1, 0.3, and 0.5 weight% of biocide mixture with a ratio of MIT-CIT 1 to 2.5 was added into this advocate paste, and it was high shear with this high shear mixture for 10 minutes before further application. After the textile screen print paste was done, the textile screen print paste was coated onto a polyester fabric using TAS Compact CX12, a textile screen printing machine. The thickness of the coating was controlled in a range of 20 to 30 micron. For the opacity test, a thin film was coated onto a Form 2A using the TQC Shin 8-side film applicator. And after the coating, the film was subjected to a data color 800 spectrometer to study the opacity of the thin film. The antibacterial testing was conducted according to AATCC 100-2019 test method, with the addition of 1000 lux fluorescent light. The test bacteria that used in the experiment will be Staphylococcus aureus. The reduction of bacteria was calculated according to the formula as below, where R is equal to the percentage of reduction, C is the bacteria recovered from the inoculant at 0 time, and A is the number of bacteria recovered from the inoculant after contact time. Let's just move on to the result and discussion of the research. 4 samples have been prepared, and the labeling of the sample was as below. XMC-@4CAT, where X is equal to number of loading of MIT-CIT biocide mixture, and MC is the MIT-CIT biocide mixture in short. Introduction of MIT-CIT biocide mixture into the system did not actually change the opacity of the system. This can be seen on the table below, where most of the sample did not change and remain at 13% for the opacity. The reason behind why opacity was studied in this research is because if high opacity of film was produced, it will limit the light penetration, inhibit the transmission of light wave, and so reduce the photocatalytic activity. So in these studies, no change in opacity means that MIT-CIT biocide mixture did not actually inhibit the light penetration, and so FOCAT can work as normal. For the antibacterial testing, it was seen that with the increase of MIT-CIT biocide mixture, the initial bacterial killing efficiency do increase. For sample 0.5 mc at FOCAT, the initial bacterial killing efficiency before 30 minutes was highest amongst all samples. However, after 30 minutes, the bacterial killing efficiency of the 0.5 mc at FOCAT was seen lower than 0.1 and 0.3 mc at FOCAT. The possible reason for this phenomenon to be seen might be due to the competition of the generated radical by titanium dioxide between MIT-CIT biocide mixture and bacteria. Studies have shown that titanium dioxide can photodegrade isothiazolinone type of biocide. This means that with radical generated by titanium dioxide degrading MIT and CIT biocide mixture, lesser radical will be available for bacterial inactivation. Hence, lower BKE was seen for 0.5 mc at FOCAT after 30 minutes. In summary, antimicrobial activity of isothiazolone type of biocide involved in two-step mechanism, which is fast bacterial inhibition that occur in minutes times, then leads to cell damaging, and so cell death after hours time. So in this current study, fast inhibition power of the MIT-CIT biocide mixture synergized with the photocatalytic activity of titanium dioxide, which improved the killing at shorter contact time. The following diagram is the proposed antimicrobial mechanism of XMC-F4CAT, where F4CAT, which is the photocatalyst, generates ROS under lights that contribute to antimicrobial, and MIT-CIT through diffusion into bacterial cell membrane, react with enzyme involved in Krebs cycle and energy generation, and so inhibit cellular activity and cell death. In conclusion, with the addition of MIT CIT biocide mixture, a new titanium dioxide metal CIT-MIT composite was successfully synthesized. With the introduce of MIT CIT biocide mixture, the opacity of the printed thin film was not affected. The initial bacterial killing efficiency of the titanium dioxide-based photocatalyst was also significantly improved. And that's all for my presentation. Thank you.